All right, good morning. Welcome to your first lesson on Punnett Squares and heredity. All right, in your first reading assigned on CK12, you read about a monk named Gregor Mendel. Mendel, sometimes called the father of genetics. Mendel was a monk in what was Austria at the time in the 1800s. One of the reasons he was a monk was because he was interested in academics and studying. And back in those days, if you were an academically inclined student, often you went to monasteries and such to continue your study, or you went into the religious orders. Now, Mendel, at his monastery, he became in charge of the garden, the monastery gardener, and in his garden he used pea plants as the organism he used to investigate these ideas and thoughts he had about inheritance. Now, we all know that children look like their parents, and Mendel sought to discover rules that we could use to determine, say, ahead of time or predict what children might look like based on the traits of their parents. So in his pea plants that he used, and again, you read this in CK12, but there was a number of traits he looked at. He looked at flower color. He looked at the size or height of the pea plant. He looked at the stems, etc. There was about 7 traits in all that he looked at. So in the last paragraph you read in that section, you were asked to predict what might happen when you take a purple plant and cross it with, I believe it was a white plant. So Mendel found that the flowers for the pea plants, and yes, pea plants can produce flowers, came in basically 2 varieties, purple and white. What Mendel found when he crossed the purple and the white plant is that all of the offspring were purple. Not a single white plant was mixed in, as long as the original purple and white plants were what we call purebred plants. So I think of dogs and breeds of dogs, right? So if you have a dog, right, like I have a dog and my dog is a mutt. My dog is part lab and part something else, so you have no idea. So a mutt you can think of as a mix of various breeds, right? So this offspring, for example, is a mutt. These offspring right here. The parents, however, were not mutts. So the purple, if we just say the flower on the left is the dad. So the purple flower here is purebred, not a mutt. The white flower here is purebred, not a mutt. With this result, and he found this also with height, tall and short plants always made tall plants. I believe it was the smooth and wrinkled seeds always made smooth seeds. So this consistent crossing, which generated one of the traits and not the other, he came up with you can imagine one of the simple laws here, what he called the law of dominance. So in the law of dominance, when 2 traits mix in a mutt, in the offspring, only one of the 2 traits is seen, and this trait we call the dominant trait. Now, the trait that is not seen is called the recessive trait. And we know the recessive trait does not disappear. It's in there, it's just hidden as the F1. Now, where we look to use these terms and ideas is and work them out really in problems is in genetic problems using the Punnett squares. So in a Punnett square, a standard Punnett square, and there are a variety of Punnett squares, but the standard one is a 2x2 Punnett square with 2 columns and 2 rows. It's a model of a crossing of 2 sets of parents and the various combinations possible in their children. So if I take the purebred purple plant mentioned previously, so we had a pure purple flowered pea plant. So what Mendel determined, or really hypothesized, and it turns out he was right, and this wasn't proven until later when we invented microscopes and we could look inside cells and see chromosomes, and we go all the way up to Watson and Crick and DNA and genes. But what Mendel realized is that most traits are determined by two factors. One we get from our mother and one we get from our father. Now we know these factors are on structures in our cells called chromosomes. So for purple, let's use a large P. So for this plant, his purple, what we call his phenotype or physical expression, his purple trait is determined by a pair of chromosomes that he got from his parents, both of which are purple because we know this plant, again, as we mentioned, is a purebred plant. The other plant in our example is also purebred but is not purple, is a white-flowered pea plant. So he or she, in this case, I guess if the other one's a he, also has a pair of chromosomes. But on his chromosomes, or on her chromosomes are small p's. So what a Punnett square does is it models sex, fertilization, and then the results in the zygotes or the offspring. When this plant right here, the pure purple plant, produces, say, a sperm, because we're making him the male, he's not going to put both these chromosomes in his sperm. In a sperm, half of those 46 chromosomes will end up in there. That's 23. In the woman's egg, half of her 46 chromosomes will end up there. That's another 23. And then the zygote goes back to the standard number. 23 and 23 gives 46. This is a special kind of cell division, not called mitosis, but called meiosis, which we'll also learn about, hopefully in school. Here we're not maintaining the chromosome number, but we're cutting it in half. So back to our example here. Of these two P's, only one of them will end up in the male's sperm. So that's rather large. We hopefully we're not. So if we imagine this is one of his sperm, this big P will end up there. The other chromosome with the other big P will end up in a separate sperm. And if we look at this female, we imagine her eggs. One of her small p's will end up in this egg, and the other small p will end up in this egg. The inside of the boxes, here, 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 and here, represent the different combinations of the male sperm with the female's egg. Now, in this case, you can see why all of the offspring will end up being the same thing. So what you have in this offspring is, the way you put the Punnett square back together is then you're going to bring this P down and this P across, this one down, and then this across, this down, and this across, and then this letter down and this letter across. So this offspring, this would be the zygote, or the baby, if you will have a big P from dad, in essence a purple gene, a white gene from mom, as will this one, and this one, and this one, and this one. So what you have in the offspring is these are all mutts. They're in essence half purple, half white on the inside in their genes. But as we discussed previously, due to the fact that one of these genes is dominant over the other, or one of these really half genes, if you will, is dominant over the other, the only thing visible in these plants would be purple. So another term or symbols really we use here as well. The parents, we use sometimes when we refer to the generations, and this was also in your CK reading, are given a P for the parental generation, and that's easy enough. And the offspring the F1 generation. And if you're taking Italian, son in Italian is figlio. So the Latin here is filial, meaning the sons and daughters, if you will. So the F1, that's not supposed to be there. Okay, so if I cross a pure purple plant with a pure white plant, or if I cross any pure dominant trait with any pure recessive trait, the offspring are all dominant. Now, this gets a little more interesting is slide. All right, we had parents on the outside making kids on the inside of the Punnett square. What happens when we cross the F1, or the children, the mutts. So let's imagine that now I'm crossing not the pure purple, but two of the mutts to each other. So a mutt purple pea plant and a mutt purple pea plant. So genetically, on the outside, the plant is purple, but on the inside, this plant is half purple and half white. Standard practice is we use the dominant trait. We use a capital letter, the first letter in the word, and we capitalize it to indicate that trait. So P for purple. For the recessive trait, which in this case is white, we don't use a W. We use the same letter we use for the dominant trait, but we use the lowercase form of it. So this plant is what we would say big P, little p. Big P, little p, excuse me. This plant is also big P, little p. Now, when we put these together in the various combinations in the offspring, we see we get more possibilities now. So as I mentioned earlier, that white trait, that white characteristic in the mutt purple plants does not disappear. It can come back in this next generation, and the generation that we're working on here would be called the F2. So the generation here would be the F2 generation in the Punnett square. The parents now are the F1s, right? So if I go back, and here those kids are now, if you will, have grown up and are producing their own kids, what we call the F2, in essence the grandchildren of the original parents. All right. So when we play our tic-tac-toe here and put everything back together, in this box, what you have is, let's leave that black, is you have a big P and a big P. In this box, you have a big P and a little p. In this box, you have a little p and a big P. But what we standard practice is to always write the dominant letter allele first, or allele is another term I'll spring on you later. And in this corner, we'd have a little p and a little p. So again, I'm gonna go through how I got those letters, and then I'm gonna erase it. And this is, in essence, what you're doing. For this box, you're combining this half a chromosome, or this part of the chromosome, with this one. So those are two big P's. For this box, you're bringing this P down and then this P across to it. For this box, you bring this P down and this P across. And then for the last box, you bring this P down and this P across. So if I look at these plants, maybe I can get fancy here with the highlighter. Maybe I'll use blue and yellow. So if we look at this plant, he has two big P's. So this plant is purple, because both his, what we call alleles, half a gene is called, the technical word is allele, A-L-L-E-L-E. Should I write that down? Allele. An allele is one letter, if you will. It's half of the set of chromosomes. So the two alleles here are both purple. Big P, purple, purple. So obviously this plant is purple. Now this plant is half purple, half white genetically. But due to dominance, this plant looks purple. If we're just looking at the outside, we're looking at the plant in a garden, we cannot tell that on the inside this plant is a little bit white. The outside, it's all purple, and similar to with this plant. However, again, that white trait, it does not disappear entirely. It stays hidden in these original mutts. And 25% of the time, those two chromosomes can come back together and generate, in this next generation, in this F2 generation, a white plant. When we report the outcomes, so you'll be asked to do crosses like this. You'll be given, say, a mutt and another mutt, or a mutt and a pure, or a pure and a pure, or a pure and a mutt, or a recessive and a recessive, asked to do a cross and determine the outcome. When you report the outcome here, if we're reporting what the plant looks like on the inside, and let's go to another color here, we would call that a genotype. The gene type. So we would report this statistically or mathematically. So the gene types here, there's 3 different gene types. There's 25% of this, 1 out of 4, you have 2 big P's. 50% of the gene types are 1 big P and 1 little p. And then 25% of the gene types are a little p and a little p. If you're asked to report what these plants look like, their physical types, that's called a phenotype, what they look like on the outside. So here, the 25% of the plants that are big P, and the 50% of the plants that are big P, little p, they're all purple. They're all purple. So it's reported, this would be reported as 75% purple flowered pea plants and 25% white flowered plants. And that's how you do a Punnett square.